Imagine a computer chip that doesn't just process data, but actually writes the code of life itself. No toxic chemicals, no complex factories, just electricity and water flowing through silicon. That is exactly what scientists have cracked open today. We are standing on the edge of a new era where biology meets electronics in the most elegant way possible. In this video, we will uncover how this breakthrough works, why it could revolutionize medicine, and how you might one day be able to rewrite your own genetic blueprint with nothing but a power source. This isn't sci-fi. It's happening right now, and it changes everything we thought we knew about the future of computing and biology. Let's dive straight into what just happened in the lab. For decades, when scientists wanted to manufacture DNA, they were stuck with traditional chemistry methods that required toxic reagents, high heat, and massive industrial facilities. It was expensive, slow, and incredibly wasteful. Then came this silicon chip developed by researchers at Harvard University, led by the brilliant Jeffrey Abbott. But here is the twist that makes this so exciting. This chip wasn't originally designed to write DNA. That was its first mistake, or rather, its most genius feature. Jeffrey Abbott started with a completely different goal in mind. He was building silicon electronics to record electrical activity inside large populations of neurons. His original design was meant to act as an external interface for the brain, reading signals from neurons without needing wires that would kill them or damage tissue. The chip utilized water-based electrolytes and electrical currents to communicate with biological cells non-invasively. It had to be safe, precise, and biocompatible because it was touching living tissue. When they tried using this same silicon architecture for something else entirely, synthesizing DNA, they realized the underlying principles worked perfectly. Instead of just reading neurons, the chip could now write genetic instructions into single-stranded DNA molecules using pure electricity and a gentle water bath. So, how does this magical process actually work? Think of it like an electric typewriter for your genome, but instead of hitting keys to print letters, you are applying voltage to push ions into solution. The silicon chip contains microscopic electrodes arranged in a precise grid pattern. When you apply an electrical current through these electrodes, it creates a localized chemical environment inside the water-based buffer surrounding them. This specific electrochemical reaction triggers enzymes that are suspended in the liquid to start assembling DNA sequences. The beauty of this system lies in its parallel processing capability. Because every point on the chip can be addressed independently via electricity, you don't have to sequence one gene at a time like in older chemical methods. You can synthesize dozens, perhaps even hundreds, of different DNA strands simultaneously right on the surface of that tiny silicon wafer. The speed is staggering compared to traditional methods, which might take days or weeks for a single complex sequence. Here, it happens in minutes. But why is this better than just using existing semiconductor technology? Because standard chips are terrible at handling biological fluids and extreme pH changes required for chemical synthesis. This custom-designed silicon platform was built specifically to interface with biology without melting or corroding under pressure. It essentially bridges the gap between rigid digital logic and the soft, fluid dynamics of biological chemistry. The result is a device that can generate complex genetic data sets in a fraction of the time it would take a supercomputer to calculate them using conventional algorithms. Now let's talk about why this matters so much beyond just being cool technology. The implications for medicine are absolutely game-changing. Think about personalized medicine right now. Currently, creating custom gene therapies or synthetic biology products for individual patients is prohibitively expensive and time-consuming. Hospitals would have to build their own mini factories just to treat one patient with a rare genetic disorder. With this chip, that process becomes accessible to any lab with basic equipment. You could potentially synthesize a custom DNA sequence tailored to a specific patient's genetic profile within hours rather than months. But there is an even deeper connection here between neuroscience and genetics. Remember the original purpose of this chip. It was meant to interface with neurons. If you can easily synthesize new DNA sequences on demand, what if those sequences code for proteins that regulate brain function? Imagine being able to tweak genetic pathways to stimulate neural growth or repair damaged circuits without invasive surgery. The fact that the same silicon architecture used to read brain activity is now capable of writing biological instructions suggests a future where our understanding of the mind and matter becomes truly unified. We might be looking at a technology that allows us to edit not just cells, but cognition itself through genetic programming controlled by electrical signals. Of course, no technology without challenges. There are concerns about the energy required to drive such precise electrochemical reactions over long periods, and the complexity of ensuring absolute accuracy in every single nucleotide added during synthesis. If a mistake occurs on that silicon grid, it could introduce harmful mutations. However, the potential for self-healing mechanisms or error-correcting DNA sequences built directly into the chip's design opens up new avenues for solving these reliability issues. The frontier is clear. We are moving from passive observation of biology to active, programmable manipulation using simple electrical commands. So what does this mean for the future? It means the line between computing and life is dissolving. We are entering an age where information isn't just stored on hard drives or cloud servers. It is becoming embedded directly into living systems in a way that was previously impossible. This technology could transform how we approach disease treatment, environmental monitoring, and even data storage itself. Imagine storing petabytes of data inside the genetic code of bacteria, synthesized instantly on demand using this silicon interface. Or consider the ability to rapidly prototype new drugs by testing thousands of unique DNA sequences against cellular models in real time, without the need for massive chemical synthesis plants. The most fascinating part is how accessible this is becoming. By leveraging a chip originally designed for neural interfaces, we have found a shortcut that bypasses years of traditional research in materials science and chemistry. This teaches us that sometimes the best solutions aren't invented from scratch, but discovered by reimagining what existing tools can do when applied to new problems. We are witnessing the birth of a hybrid discipline, where engineers and biologists work together seamlessly, using electricity as the universal translator between machines and life forms. As we stand on this precipice, one thing is certain. The future belongs to those who understand how to speak both the language of silicon and the language of biology. The ability to write DNA with electricity gives us a powerful new pen to reshape our world. Whether it's curing diseases, creating sustainable energy solutions through bioengineering, or unlocking the secrets of consciousness itself, this technology provides the foundation for all of it. We are no longer just observers of nature. We are becoming its architects.